Hello guys, this presentation handling over by Hari Tiridesh, Yashwant, Jayakash, Samir, Trisha, William, Medarnav, Manasa, Sneha and Ram Tejesh. Now we are going to see about the topic DNA.
Basic structure of DNA. DNA or deoxyribonucleic acid is a molecule that carries the genetic instruction used in the development and functioning of all known living organisms. Genetic code. It contains the DNA genetic code that determines the characteristics of an organism. The sequence of nucleotides, adenine, thymine, cytosine, and guanine, forms the genetic information. Double helix structure. The DNA molecule has a distinctive double helix structure discovered by James Watson and Franz Crick, with two strands winding around each other. Role in heredity. DNA plays a crucial role in heredity, passing genetic information from one generation to the next. It carries the instruction for building and maintaining an organism. Replication and protein synthesis. DNA undergoes replication during cell division, ensuring the transmission of genetic information. It also serves as a template for synthesis of RNA, which in turn is essential for protein production in cells.
DNA replication. DNA replication is the process by which a double-stranded DNA molecule is occupied to produce two identical DNA molecules. Replication is an essential process because whenever a cell divides, the two new daughter cells must contain the same genetic information or DNA as the parent cell. The replication process relies on the fact that each strand of DNA can serve as a template for duplication. DNA replication initiates at specific point called origins, where the DNA double helix is unwound. A short segment of RNA called a primer is then synthesized and acts as a starting point for new DNA synthesis. An enzyme called DNA polymerase next begins replicating the DNA by matching bases to the original strand. Once synthesis is complete, the RNA primer are replaced with DNA, and any gaps between newly synthesized DNA segments are sealed together with enzymes. DNA replication is a crucial process. Therefore, to ensure that mistakes or mutations are not introduced, the cell proliferates the newly synthesized DNA. Once the DNA in a cell is replicated, the cell can divide into two cells, each of which has an identical copy of the original DNA.
Gene transcription. Gene transcription is the process where a gene's DNA sequence is used to synthesize a complementary RNA molecule. Initiation. RNA polymerase binds to gene's promoter region, signaling the beginning of transcription. Elongation. RNA polymerase moves along the DNA template, synthesizing RNA in the 5 to 3 degree direction. Template strand. One strand of DNA acts as a template for RNA synthesis. Determine the sequence of the complementary RNA. Coding strand. The other DNA strand has a matching sequence of the RNA, except with the uracil U replacing by thymine T. Termination. Transcription concludes when RNA polymerase reaches a termination sequence leading to RNA release. The RNA processing. In eukaryotes, pre-mRNA undergoes splicing to remove introns and add a 5 cap and 3 poly A tail. mRNA export. Processed mRNA is transported from the nucleus to the cytoplasm for translation. Translation. mRNA serves as a template for protein synthesis at the ribosome, following the genetic code. Regulation. Transcription is highly regulated, influenced by various factors, ensuring proper gene expression in different cell contexts.
Translation. Definition. Translation is the process by which the genetic code contained within a messenger RNA molecule is decoded to produce a specific sequence of amino acids in a polypeptide chain. It is performed by ribosome. Initiation. The ribosome assembles around the target mRNA. The first tRNA is attached at the start codon. Elongation. The last tRNA, validated by the small ribosomal subunit accommodation, transfers the amino acid. It carries to the large ribosomal subunit, which bind it to the one of the preceding admitted tRNA, transpeptidation. The ribosome then moves to the next mRNA codon to continue the process translocation, creating an amino acid chain. Termination. When a stop codon is reached, the ribosome releases the polypeptide. The ribosomal complex remains intact and move on to the next mRNA to be translated.
Genetic mutation. Genetic mutation is a change in the sequence of DNA that makes up a gene. These changes can occur naturally or be induced by external factors like radiation or chemicals. Types. Mutation can be classified into several types, including point mutation involving a single nucleotide, insertion, deletions, duplications, and chromosomal rearrangements. Causes. Mutation can result from errors during DNA replication, exposure to mutagenic agents, or even be inherited from parents. Some new mutations have minimal impact, while others can lead to genetic disorders or contribute to development of diseases like cancer. Effects. Depending on the location and nature of mutation, its effect can vary. Silent mutation may have no observable impact, while missense or nonsense mutation can alter the functions of the protein encoded by the gene. Evolutionary significance. Mutations are essential for evolution, providing the genetic diversity upon which natural selection acts. Beneficial mutation may confer advantages, leading to the survival and reproduction of individuals with those mutations, while deleterious ones may be eliminated from the.
Inheritance. Genetic material. DNA or deoxyribonucleic acid carries the genetic information and it is located in the nucleus of the cell. Chromosome. DNA is organized into a structure called chromosomes. Humans typically have 23 pairs of chromosome with one set inherited from each parent. Gene expression. Gene segments of DNA contain instruction for building proteins. The combination of gene inherited from both the parents influence trait and characteristics. Alleles. Different versions of a gene called alleles. Individuals inherit one allele from each parent, contributing to genetic diversity. Mendelian inheritance. Inheritance follows Mendel laws, including the law of segregation. Alleles separate during gamete formation, and the law of independent assortment. Genes on different chromosomes assort independently. This results in diverse genetic combination in offspring.
DNA profiling. DNA profiling is a process where a specific DNA pattern called a profile is obtained from a person or sample of bodily tissue. Sample collection: blood, buccal swabs, hair, bone with fingernails, tissue from internal organs, including brain, muscle, and skin. DNA extraction: DNA is extracted from the specimen, that is, blood, semen, skin, hair. After DNA extraction, the enzymes are added, which work like scissors to cut the DNA into smaller segments that are different between individuals. DNA amplification. Application of the template DNA is achieved by repeating a 3-step process through 25 to 30 cycles in a thermal cycler. Each cycle has 3 steps. The 2 DNA strands are denaturated by heat. The sample is then cooled to allow the primers to anneal to the DNA segments. DNA analysis. Human DNA profiles can be used to identify the origin of a DNA sample at a crime scene or testing for percentage. DNA profiling is used to identify the probable origin of a body fluid sample associated with the crime or crime scene. Reveal family relationship, data comparison and interpretation. The Combined DNA Index System (CODIS) blends computer and DNA technologies into an effective tool for comparing DNA profiles. The current version of CODIS uses 2 indices to generate investigative leads in the crime, where biological interpretation of DNA typing results depends not only on population genetics but also on laboratory error. 2 samples might show the same DNA pattern for 2 reasons. 2 persons have the same genotype at the locus studied, or the laboratory has made an error in the sample handling procedure or interpretation.
Genetic engineering and its definition. Genetic engineering involves manipulating an organism DNA to introduce specific traits or characteristics. Tools and techniques. Scientists use various tools such as CRISPR or Cas9 to precisely edit gene by adding, deleting or modifying DNA sequence. Application. Genetic engineering is employed in agriculture to create genetically modified crops with improved traits, in medicine for gene therapy to treat genetic disorders, and in industry to produce valuable substances like insulin. Ethical considerations. The practice raises ethical concerns including potential unintended consequences, environmental impacts, and questions about the morality of altering the genetic makeup of organisms. Regulation and oversight. Many countries have established regulation to govern genetic engineering to ensure responsible and safe practices, addressing both ethical and environmental implications.
DNA computing. DNA computing leverages the parallel processing capability of DNA molecule to perform multiple computations simultaneously. This is due to the massive parallelism inherent in the biological process of DNA replication and transcription. Massive data storage. DNA can store vast amount of information in a compact form,
DNA computing explores the potential of using DNA as a medium for information storage, allowing for dense and efficient data representation. Computational density. DNA based computation can achieve high computational density by encoding information in the molecular structure. This enables the representation of complex data in a small physical space, offering advantages in terms of scalability. Biological inspiration. Inspired by biological process, DNA computing harness principles from genetics and biochemistry. Algorithms are designed to mimic the way DNA naturally processes information, providing a unique approach to solving computational problems. Non-binary logic. Traditional computers use binary logic 0s and 1s, while DNA computing introduced non-binary logic. DNA molecules can represent multiple states, allowing for development of novel algorithms that exploit this richness in information representation. Potential for parallel evolutionary algorithms. DNA computing can be applied to implement parallel evolutionary algorithms, enabling the simultaneous exploration of multiple solutions to complex problems. This mimics the natural evolutionary process found in biology and can be beneficial for optimization tasks.
Here is a conclusion. Structure of DNA is important in many events in cellular life. Replication, transcription, and regulation of expression of many genes depends on local difference or changes in DNA structure. Recombination, which leads to rearrangement of gene, takes advantage of the ability to form an unusual structure called a holiday structures. Also, different kind of mutation occurs as a result of specific DNA structure. Thank you.